Good evening, everybody. Welcome to our last speaker in the series of the Dean's Seminar for the spring semester. I really want to thank our faculty for stepping up and sharing with us their work that they're doing. It has been absolutely inspirational to hear every single person's presentation, and I think we're in store for a wonderful talk by Dr. Mark Miller. Mark is an assistant professor in the Department of Kinesiology. He has a very interesting background and training. He is the first that I've ever met who is or has had an aerospace engineering undergraduate degree, as well as having completed a mechanical engineering degree from the University of Vermont for his doctoral degree. He combines his engineering experience with the field of kinesiology to be a really innovative and translational researcher. He works on understanding the factors that influence muscle function at the molecular and cellular level, and then designs interventions in humans that can ameliorate the detrimental effects of aging specific to sex. He has multiple active grants. He's a PI and he's also a co-investigator. He's very prolific in his scholarly products. And he's really devoted to teaching, and he's been amazing in how he's attracted students and has been working with students in his lab. So Mark, thank you very much for volunteering, and we look forward to your talk. Oh, but I forgot one thing that he shared with Carrie during the opening remarks. He's actually started bike riding. And so I think we should all strive to do what Mark's been doing with his 12-year-old son of increasing the number of miles he can do in a trip. And he's now up to 30 miles per ride. So pretty impressive, Mark. If you get up to your goal of 50, I might be able to maybe have you come to my house and I'll offer you a drink with your son. So without further ado, Dr. Miller.
All right, well, thank you for that introduction and thank you all for coming. I can see a subset of you, so I'm going to tell you the same thing I tell my undergrads. If you're comfortable turning on your camera, it's great to see faces, but if you're not, it's fine to leave it off as well. And please feel free to stop me as we go. If there's a question that comes up, don't hesitate to stop me because this is going to build, and I don't want you to get lost or have questions that are lingering for too long. So, to get started here. So we're studying aging and skeletal muscle, and why do we care about this? Well, this is a poll that was taken back in 2018, looking at what worries people about aging. You can see here the two biggest concerns are losing mental function and losing physical function. Even more so than dying, which was only ranked at like 26%, or losing looks down at 15%. So declining health is a top fear Americans have about getting older, and there's a good reason for that. As we age, we start to see all kinds of difficulties. So about 20% of adults in their 50s have limitations in activities of daily living. And what I'm talking about there is walking, climbing stairs, rising from a chair. And as we age, this gets even worse. So 40% of adults older than 80 have this problem. And there's sex-specific effects here. So older women are more likely to experience functional limitations and disability. And this isn't just because women live longer. If you take that into account, women just have more disability. And this sex-specific difference increases as people get older. And so one of the things we can look at is muscle power. And so muscle power, I'm going to talk about this a lot throughout this talk. So muscle power is the product of force times velocity. And it's a key predictor of this age-related decrease in functional capacity. So as we age, our functional capacity goes down as our muscle power goes down. And so how do we study this? Well, in my lab, we try to take an approach where we look at a variety of anatomical levels. And so today I'm going to focus on muscle function, and we try to look at it, like I say, at all these different levels, which makes us somewhat unique. We start at the whole body level, and we do things like activities of daily living. That picture there is a gentleman doing a chair rise test. They stand up from a chair 10 times, and then we measure how fast they can do that. We also do dynamometry, which I'll explain a little bit more as we go along. This allows us to look at specific groups of muscles, and then we do biopsies. So we look at the single fiber level, and then we can also get down to the level of the myosin actin interactions. This isn't going to be a focus of my talk today, but we also do this with muscle structure. So we start at the whole body level. We do things like dual energy X-ray absorptometry, or DEXA, and we look at whole body fat, whole body lean mass. We also do here, you can see a magnetic resonance image with white here is fat. Then you can see the muscle in here. This is work we do with collaborators. This is work of Dr. Jen Kent's lab. We also do things at the single fiber level. So your muscle is made up of 3 different types of myosin. A myosin that contracts slowly, one that contracts fast, and the one that contracts very fast. And we can separate these on a gel and look and see how these things are changing. We can also do various things with immunohistochemistry and various staining. What you're looking at here is the fibers are basically coming out of the screen at you. And with different chemicals, we can stain the different myosins. So a green means it's an MHC1 or slow contracting myosin. A red means it's a fast contracting myosin, or an MHC2A. And then MHC2X are stained blue. And so when you have a mixed fiber type here that is expressing both 2A and 2X, you get this purplish looking fiber. We can also do things such as staining for lipid droplets. This is an oil red O stain, so all the red droplets you see there are fat droplets. And we can look at size and number and things like that. We also go down to the molecular level, or the excuse me, myofibrillar level, where we can look at things like the dark bands here, the thick filaments, Z bands, and things like that. So we get very low at the structural level. What the lab is really good at, and what we focus on, is what happens here at this molecular and single fiber level. And so how do we do that? Well, we do biopsies. So we do typically thigh biopsies. So this is the vastus lateralis. And again, I'm saying we, but we do this with a collaborator. Dr. Stuart Chipkin comes and does these biopsies for us. And then we take some of this tissue, we freeze it, so we can look at proteins. Some of it we prep for that immunohistochemistry, those colors that I showed you. But some of it we bring back to the lab and we cut into bundles like this. And if you look closely, you can actually see the fibers running vertically here. You can see some droplets here. These are fat droplets. The red is some blood that's still left in that sample. And what we do is we take these various chunks of tissue that we cut up and we tie them to glass rods and we stretch them a little bit. That way they don't contract and turn into a ball and they become unusable. And so this is all done the day of the biopsy. When we want to do an experiment, what we do is we go to the freezer, we take these out, and then we pull individual fibers. And so what you can see here on the left, this is a muscle fiber, a human single skeletal muscle fiber. And to put these on our rig, we have to put these T-clips on the ends. And these are basically a donut with a long flat piece, and we wrap that flat piece around the fiber and we crunch it down so we really have a good grip on that fiber. And then we take these fibers and we put them on our rig. So we've got a hook here that's a length hook, and a look here that's a force transducer. We put the fiber on there, and then we can measure force and we can change the length. And if you back up a little bit, you can see this is what our rig looks like with all these different chambers, with all these different solutions, so we can dunk this fiber in various solutions and see how it responds. One of the nice things about this prep is we can monitor sarcomere length throughout the experiment, and so we can keep that constant, keep that at the values that we want. And so coming back to aging, what we want to do is look at all these different levels and see what happens with aging. And the goal behind this is that if we understand the mechanisms behind the age-related power loss, this will help us develop countermeasures, either drugs or exercise. And again, I'm going to focus on this power aspect, which is force times velocity. And so what do we know so far is that as we age, we know our muscles get smaller. And so we get a loss of muscle quantity. We get muscle atrophy as we age. The other thing we see is if we take some of this tissue from a young and an older adult, some studies have shown a decrease in force or a decrease in velocity. So this is a loss of muscle quality. And really the focus of our experiments here no one had looked at molecular interactions at this point. So we were wondering, are these molecular interactions as myosin and actin affected by age? And if so, can these altered interactions explain the reductions in whole muscle power output that people have seen in the past? I do want to point out that muscle contraction is derived on a variety of different factors. And so all these different things go into muscle contraction. We're just focused on skeletal muscle contractile properties, but there are a number of things that actually can change muscle contraction. And any of these factors could be altered potentially with age. And so I want to spend a few minutes orienting you to myosin and actin and how this works. So down on the bottom, you're seeing a video of myosin as it's taking in ATP, which is the energy source. It's turning it into ADP and phosphate. It's binding and going through its power stroke and doing what myosin does. One way to do that is to draw this up here. And you can see this is a diagram of time on the x-axis and force on the y. So myosin is off for a long period of time, then it binds, it generates force, it stays attached for a period of time, and then it comes off. And myosins typically are off for about 80% of the time, and they're bound on for about 20% of the time. So in your muscles, you have hundreds of thousands, millions of these myosins binding and causing our muscles to work in the way that they do. I don't have a room here to draw that many myosin, but let's make a muscle out of 10 myosin. So here's 10 myosin heads. As I told you, they're typically bound for 20% of the time. So you see this first head is bound from 0 to 20% of the time. This one 20 to 40, 40 to 60, and so forth. So what this shows is that if you have 5 myosin heads, you'll have 1 myosin head bound at each point in time. Now I've drawn 10 heads here. So if you just draw a line through any point in time, you can see that the force that's generated by this fiber I make is 2 myosin heads. As the dean said, my background is in engineering. This requires me to draw this as an equation. So here you can see this is isometric force, so the force we're generating. It's the amount of time that the myosin is bound over the total cycle time, which I told you was 20%. So that's this 0.2. The number of heads that are here, which is 10, and then the force per crossbridge. So this is, if we plug the numbers in, you get 2 here, just like you can visually see. But the force this fiber is generating is the force of 2 crossbridges. So one of the other thing that is important, as we talked about with power, is velocity. And so velocity is proportional 1 over the attachment time. So what do I mean here? We've got myosin heavy chain 1 fibers, which contract slow, which means their myosin stay attached for a long period of time. And so it's a little hard to do on Zoom here, but the way I like to think about it is if you're grabbing a rope and you're pulling it, and you're grabbing and holding for a period of time, that rope's not moving very fast. As you start to grab and let go more quickly, that rope moves quicker. And the faster you start to grab and let go, the faster that rope's going to move. And so when you are holding on for a long period of time, your velocity is slow. For a moderate amount of time, your velocity is fast. And if you're holding on for only a short period of time, then you've got a fast contractile speed. And so that's where we come up this velocity is proportional to 1 over myosin attachment time. And so what some people have seen in the past, this is a study by D'Antona, is that they found that the force generated, so this is force, you'll see this throughout the talk, a tension, which is a force per size. You'll see that the force generated per amount of myosin, you get more force as you get more myosin. So here's a slow contracting fiber, MHC1. A young muscle has lots of myosin, so it's got higher force. As you get older, they found that the myosin went down, and then they did older immobilized, and they see that it goes even lower. And then they see the same thing for fast contracting fibers. And so we can draw this as, let's say we just reduce the number of heads by 20%. So we've done that here. So we've gone from 10 heads down to 8. So what we can see here is that the force is going to drop by 20%, which isn't surprising. We're dropping down to 1.6, but our velocity is going to stay the same. So this is one way the hypothesis here, this is one way that aging could decrease single fiber force. The other thing that could be changing with this myosin actin crossbridge kinetics is not just losing myosin, but what happens if you change the way that these myosin and actins interact. So here we're going to increase myosin attachment time. So the dashed line is where we started. The solid line is now we're increasing that myosin attachment time by 20%. What you can see here is that now you only need 4 myosin to have one bound at any point in time. So if we plug this into our equation, not surprisingly, force goes up. Okay, you're generating more force if the myosins are attached for a longer period of time, but you're decreasing your velocity. This could be a way that aging is decreasing your single fiber velocity. So we'll look at these two, think about these two in the back of your head as we're going through the data, and we'll see which one of these, or if any of them, pan out. So again, I'm going to step back here. We're going to start at the whole body level and work our way down for one of the studies that we did with aging. And so what we wanted to do here is we wanted to recruit people that had similar physical activity levels. This was very important to us. As we age, we know we start to become more physically inactive, but we didn't want to have that as a cofactor in our study. We wanted to match these people for physical activity levels. So they wore accelerometers for a week and we made sure they were the same. What became interesting about this though, is we recruited the young adults first. And these are 21 to 35-year-old people. And we recruited them if they were sedentary, which means less than 2 sessions of 30 minutes or more a week. Turns out sedentary young adults are relatively active compared to older adults. So the older adults we recruited were moderately physically active. So 3 to 5 sessions of 30 minutes or more a week. And this is typically done in walking or hiking. And then all these volunteers were healthy in a variety of measures that we looked at. So what happened? So here we've got our 4 different groups. We've got 5 young men and 5 older men, 7 young women and 7 older women. And just to let you know, this was not the initial design of this study. The idea was to have equal numbers of men and women in a young group and an older group. So this was supposed to be young versus old. And what happened was, as I was going through the data, it became very clear that men and women were responding differently. Now, you may look at this and say, well, those are really relatively small n's, and they are. But part of that is because we're collecting so much data, we can only put so many people through the study. But since our study design was 12 and 12, and we look through the data, suddenly we see we have to divide into 4 groups instead of 2. You can imagine the stress level might have gone up a little bit to say, uh-oh, are we going to be able to see differences here with such small groups? But we did. Things turned out just fine. And so the young group was around 24, the older group was around 70, but we did match well for physical activity. There were no differences there. But if we look at peak oxygen consumption, so this is a measure of aerobic capacity, we did see it went down in both the men and the women, even though we matched for physical activity. You're going to see a number of these graphs, so I want to orient you to these. The men are going to be on the left, the women are going to be on the right, and the open is the young and the closed is the older. So you can see here for both men and women, their aerobic capacity has gone down, even though they're matched for physical activity level. And so coming back to our chart here, what we see is no change in physical activity, but we see a decrease in functional performance in terms of VO2 max. So the next step is to go to the whole muscle level. So I'm going to show you a little video here of dynamometry in case you haven't seen this before. And the way this works, we put you into a machine, you're strapped in this chair. We're looking at the quadriceps muscles or your thigh muscles. And at first you're going to see someone pushing against the machine and the machine isn't going to move. So this is an isometric contraction. So they're going to push as hard as they can and generate a lot of force. Then we're going to allow the machine to move at different speeds. And you'll see 2 speeds here, a fast speed and a slow speed. And this allows us to get out muscle power because now you have a velocity. You'll also hear someone cheering this person on, which is what we do in these studies. We just don't sit there quietly and say nothing. We want them to do their max, so we tend to push them a little bit. All right.
Kick, 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 kick. Keep it up, keep it up, keep it up, keep it up, and stop. Nice. Kick, relax. Kick, relax. Kick, relax. Nice. Kick, relax. Kick, relax. Kick, relax. Nice.
All right, so that's what our dynamometry looks like, and that's about what we do. If we're doing a one that they're allowed to move, we do 3 contractions at a time. And so for this study that I'm talking about today, what we did was we locked people in 4 different positions. The machine doesn't move. You kick against it as hard as you can, and you get a measure of force out. And so here we've got torque, which is a measure of force, versus the different knee angles. And what we saw for our 4 different groups was where there were no differences. So whether young or old, men or women, everything came out the same. Now we're going to allow this dynamometer move, and it moves at a specific velocity, either slow or fast. And what we can get out now is power. So this is power versus speed. So slow speed on the left, high speed on the right. And what you can see here is the open circles are the older men, and they decrease with age. So the closed circles are the older men. And then the women, you can see, have a much larger decrease in power output than the men. And so aging decreases whole muscle isokinetic power production, and especially so in older women. And so we can add this to our charts. We've got no change in force, but a decrease in power. So now we're going to start looking at things at the single fiber level. Excuse me. I somehow, without going out of my house or going anywhere, I've caught a cold this week. So as I talked about before, if you got these 3 different myosin heavy chain isoforms in your muscles, and the slow myosin heavy chain 1, you have slow myosin heavy chain 1, one contracts fast is MHC2A, and one that contracts the fastest, which is MHC2X. And we can run these samples on gels. So we separate out the different isoforms, and we look at these different people, because if suddenly the older people had a lot more myosin heavy chain 1, that could explain why they have slower contractile velocity. Well, we didn't see any changes. So when we match for physical activity level, we see no changes in this myosin expression. We also looked at the amount of myosin that's there, as well as the amount of actin, since these are 2 very important proteins in muscle. No changes there either. And so if we step back to our hypothesis that the decrease in force with aging might be due to these loss of myosin heads, we didn't see that. We saw no loss of myosin. And this may be because we're matching for physical activity level. So now we start looking at the fiber type information. So this is maximum tension. So again, this is force per size. So this is a measure of force in slow fibers and in fast fibers. And what we're seeing here is an increase in the ability to generate force in the older women. So the force generating power of the older women is going up. When we look at this, if we try to compare force generation with their torque generation, there's absolutely no relationships in slow or fast fibers. So the amount of force that they're generating in the fiber doesn't seem to be turning into what's going on with the torque. But what is kind of interesting is when we look at this force, we do see a correlation with the amount of power that they can generate. So the more force you're generating, the less power you're generating. And so we can add that onto our summary here, is that we're seeing this increase in force, which is correlating well with this decrease in isokinetic power. Well, hopefully at this point, you're kind of scratching your head and saying, wait a sec, you're seeing an increase in force, but a decrease in power, which is the same thing we did. It's like, what's going on here? How can this be happening? That doesn't really make sense. So we kept digging and going deeper and deeper. And so if we start to look at myosin actin crossbridge kinetics, we've kind of talked about these before, but there's 2 different measures we can get from what we do. We can get this rate of force production, which is how fast myosin goes from weakly bound to strongly bound. And we can also find this, how long myosin has been attached, okay? So if we look here at the slow and fast fibers again, we can see that the rate of force production is going down more in older women than it is in men. And so men aren't changing with age. Women's crossbridge kinetics are getting slower. And if you look at myosin attachment time, we see in the slow contracting fibers, there's really no change. But in this case, both men and women are getting slower with age. And what's we thought was kind of exciting is if we look at these myosin actin crossbridge kinetics, they correlate well with how much power people can produce. So as the crossbridge kinetics get slower, the isokinetic power becomes less. And same thing here. This side means the crossbridge kinetics are getting slower, and therefore their power is getting less. So when we look at this and kind of try to put it all together in the end, we're seeing that these slowing of crossbridge kinetics cause an increase in isokinetic or isometric tension, but this is causing a decrease in power. So what's missing here? Well, when the myosin's binding and staying attached for a longer period of time, like I talked about with the rope scenario, you should be seeing a slowing of contractile velocity. So that's what we predict is going on. At the time, we couldn't measure that. Now we can, but at this point in time, we couldn't measure it. So that we think this is predicted. This is just correlated because it's got the same underlying myosin actin kinetics behind it. And so overall, crossbridge kinetics, we think, are potentially a clinically relevant measure that can explain the loss of whole muscle performance, and especially in women, because we're seeing larger decreases there. So the next step is to see if we can reverse this. And I have some data on that, but before I did that, I wanted to pause here for a moment and see if anybody had any burning questions before I go on.
Mark, I have a question. As a nutritionist, I wonder if there are any differences among the individuals in the study based on, let's say, protein intake that we know can sometimes decrease with age, especially among women.
No, it's an excellent question, and we did not do that in this study. We have now been working with someone who's a nutritionist down in Yukon, and we're trying to get some studies with them. And so for some of the studies moving forward, we're going to start having them do a dietary log and some different information like that. But for this study, we did not do that. But it's an excellent point that nutrition can play a vital role in all this. Were there any? Yep.
Hi, it's Laura.
Hi.
I was surprised, I guess maybe because I'm an endocrinologist, not to see any sex or age related effects on either myosin or actin levels in I don't know, that sort of surprised me. Although I guess on the flip side, a lot of us use these as loading controls when we run Western blot. So I guess maybe we should feel relieved about that. But my question is a technical one, which is, what is your control? Where, you know, a lot of times people use actin or myosin, or they use those as housekeeping genes. So how do you control for differences sort of just in protein level or muscle level when you're evaluating that?
Right. No, it's a good question. So one of the ways we do it is that, so typically when you put, run something through a gel, you're doing it on the amount of protein, right?
Mm-hmm.
So you load a certain amount of protein at the top of the gel and then you run it. But in order to look at the amount of myosin and actin that's there, it becomes a bit more complicated. What we do is we take single fibers, we put them on a rig, and then we measure the cross-sectional area and the length of that very precisely. And then we run those fibers on a gel by volume rather than amount of protein.
Mm-hmm.
And so you put a certain amount of volume in each well. And so if the myosin's darker in one and lighter in another, you know that that's changed in that way.
And are there any like post-translational modifications that could be present that still would be sex specific, that wouldn't be detected by the resolution of your gel?
Absolutely. Yes. Post-translational modifications are a big deal. I didn't include that in this set of slides, but in this study, we actually looked at regulatory light chain phosphorylation. So the regulatory light chain is a protein that binds to the myosin head, and when it's phosphorylated, the myosins pop off the backbone and are more ready to bind. And when the phosphorylation gone, the myosin heads are more closer to the backbone. And what we saw in the older women was a reduction in this regulatory light chain phosphorylation, and it was only in the older women.
Cool.
And so that's one of the mechanisms that we think might be going on. But as you probably well know, there's so many post-translational modifications. Trying to decide which one to look at and how to look at it can be sometimes a daunting task, but that is certainly something we're very interested in, is how are the proteins modified once they've been in the body for a while,
Awesome.
or with aging.
Thanks, Mark.
Sure. Any other questions? All right. So the next step was then to start thinking about how can we reverse this. And so our thought was that maybe we can do this with exercise. And is there any suggestion out there that the sexes respond differently to exercise? So again, the study that we just did, and we found all these differences by sex, that wasn't the initial plan. We weren't expecting to find that, but we did. And so if we start looking at exercise to improve whole muscle power, one of the things you can do is traditional resistance training, which is high load, low velocity, or 80% of 1 rep max. And just to be clear, 1 rep max is the amount of weight you can lift just once. So you can only lift it once and then you can't lift it again. So if 100 pounds is your 1 rep max, 80% of your 1 rep max, you're exercising at 80 pounds. And if you do this type of training, not surprisingly, this primarily improves force production. Or if you do something like power training where you're lifting 40% of your 1 rep max, so you're doing high velocity, low load, this tends to increase both contractile force and velocity. And as age-related deficits are more based on changes in velocity, power training should be more beneficial. But a number of studies that have been done said that resistance and power training had the same improvements, and they predict conflicting results in terms of functional performance. But I think what's interesting here is that a potential problem is these studies were taking men and women and lumping them together as a single cohort, even though there is other evidence out there that men and women respond differently to the same exercise. And so there is evidence out there that's saying resistance training increases overall muscle size, so this muscle quality, and power output more in men than women. And if people that have done biopsies on this, there's only been a few, see this. So that this is a study by the Trappies a number of years ago, looking 1 paper's in men and 1 paper's in women, well matched for age, same number of folks. You can see here that for the men, fiber size increases a lot, velocity increases a lot, power increases a lot. Same study done in women, there's almost no changes. Now, it's not all bad news. If women exercise for a longer period of time, like a year, you can then start to see some of these different changes. But the other good news too is that power training appears to increase muscle quality more in women than men and improves muscle power more than resistance training. And so this is what we kind of wanted to start thinking about is, are these different exercises going to respond in men and women differently? And that's the focus of the study we're doing right now that I'll get to in a minute. But I did want to show you some data that we've seen that exercise does affect men and women differently. And so this is a set of experiments that we did in people with knee osteoarthritis. So they have knee pain if they exercise too hard. So this is a moderate load, so it's only 60% of 1 rep max. And this is in MHC1 fibers, or slow contracting fibers. And what we're looking at here is a power versus force. And the solid lines are the pre, and the dashed lines are post. And you can see the men in black and the women in gray. And what hopefully is striking to you is that if the men exercise, they move to the right on this curve. As the women exercise, they move to the left. Same exercises, they're doing the exact same thing, but all the way down to the fiber level, they're responding in different directions. So we can look at here, this is the maximum tension here. So you can, if you look at this data, there's this training by sex interaction. Men are going up, women are going down. The optimal tension is where max power occurs. Again, men are going up, whereas the women aren't changing. And so what's driving this? Well, if we take a closer look, we can see that the number of myosin heads that are bound are going up in men, where they're going down in women. And this myosin attachment time that I talked about is going up in men, and it's not changing in women. And if we start to look at, the changes in these number of heads that are bound with this change in time on, we see this nice relationship here. That is, if you're increasing your time on, you're getting more heads bound. And so in general, what we found here in these slow contracting fibers is that in men, this higher force production after exercise was caused by an increase in the number of heads that bind. We also saw sex-specific effects in faster type fibers. So this is a mixed fiber type. As we age, we start to express more and more of these mixed fibers. So this is a 2A and 2X fiber. Again, we've got power on the y-axis here, but now I've put velocity on the x. Same thing that happened last time. You can see that men going from the solid to the dash shift to the right, the women going from the solid to the dash shift to the left. And so if you look at the optimum velocity, this is where peak power is occurring. Men's velocity is going up, women's velocity is going down. And what became interesting here is that the amount of MHC2X, or this fastest contracting isoform, when exercised got more in the men, but it really didn't change in the women. And what we see here is that this myosin attachment time correlates well with how much of this fast myosin you have, which this is a correlation we'd better see. If we didn't see this, we'd be really surprised, because this is how we understand things work, but it's nice to see it come out. In that the more fast isoforms you have, the shorter myosin attachment times you get. And so basically the men are responding to this exercise in a way that makes their velocities faster, and the women aren't. So again, this was resistance training. We didn't have power training. So that was the next study we embarked upon, is looking at the power versus resistance. So this is the CHAMP study. So we call it CHAMP. This is the Cultivating Healthy Aging and Muscle Performance. A little over a year ago, we were starting our 4th year of this study. This study has now obviously been halted because it's in older adults, but we're hoping in the near future, with the number of vaccinations going on in older adults, that we can start this study up again. But what this is focusing on is the question that we've been building to is, what's the difference between resistance and power training at these different anatomical levels in men and women? And so for resistance, we're doing 80% of 1 rep max. And for power training, we're doing 40%. We're hoping to get in 40 volunteers, half of which are women, and they do this for 16 weeks. And what's sort of unique about our study is we've got one leg undergoing resistance training while the other leg undergoes power training. And so everyone's their own control. So if I brought one person in and had them exercise resistance and one person exercise power, there's lots of differences, you know, your nutrition levels, your genetics, all these different things. So in this case, everyone's serving as their own control. The participants will sometimes ask if that's going to cause problems. And I jokingly tell them, yes, you'll walk in circles for a while, but you'll be okay after a few months. But no, there's no problem with exercising the 2 different legs in different ways. It's not going to cause problems in these older adults. And so I won't go through all that we're doing, but we are taking measurements at all these different levels to try to understand what's going on in these participants. And what's great about this study is that we've got over 50 visits that people have to come to the university for. So 48 just for exercise. And then we've got 4 pre-exercise and 4 post, where we're doing blood work and biopsies and MRI scans and all the different things that we need to do. And what's great is they only have to go to one location at this point. So the volunteers go to the Institute for Applied Life Sciences. They park in there and they walk in the door. They stay on the same floor to do everything, except they have to go down a set of stairs to do the MRI and come back up. But besides that, everything's done there. Now the tissue we bring back to our lab in Totman and do all of our work there. But the facilities at Isles have been fantastic for us in order to get people there and get them excited about what we're doing. This is something I stole from Professor Vandenberg the last time I saw her present a few weeks ago. I thought it was nice, since these talks are focused on undergrads, was showing us some of the different undergrads that have been in the program the last few years. Our numbers are down right now because, again, the studies are closed and it's hard to get people in the labs, but hoping to ramp back that up again as we move forward. So I certainly want to thank all the volunteers that we have, who literally gave a piece of them for these studies. Excuse me. Sorry about that. And certainly the folks in the muscle biology lab, all the people that are in italices here are the undergrads in the lab, our collaborators at the University of Massachusetts, as well as at the University of Vermont. And then our funding sources from the National Institutes of Health, and we've also gotten funding from the Human Magnetic Resonance Center here at UMass. So that kind of wraps up what I had to say. I'm ready to take questions.
Hey, Mark.
Hey, Chris.
Nice talk. I really enjoyed that. I just had a couple questions for you. So one, when you say that power exercise at 40%, how many reps are they doing for that? Because I know sometimes when you go lightweight, you do a lot of reps, but typically power work is focused on high speeds. So you try not to do too many repetitions.
So we are matching these people for the amount of work done. So for the resistance training, it's 3 sets of 8 repetitions because it's 80% 1 rep max. So because we cut the 1 rep max in half, we double the amount of repetitions. So it's 3 sets of 16 repetitions. So the idea there is we're matching for work.
Okay. And is the emphasis there still speed during those contractions?
For the power, yes. Yes, we're telling them to kick as hard and fast as they can without clanging the machine or doing things like that. And then the idea of the resistance training is you're taking 1 to 2 seconds for them to come up and then nice and slow back if you're doing like a leg extension. So yes, the idea is to do it as quickly as you can for the power part of it.
Okay. And then my other question was on some of the first stuff that you presented, where you showed the differences in power output and force with the older women. The measures on the biodex, were those normalized to muscle mass? And if not, what role do you think that plays in kind of what you saw?
Yeah, so those were normalized to lean mass. We used a DEXA machine where we could separate the legs and do it that way. So yes, I didn't say that, but yes, I should have. Those are all normalized to muscle mass, or else you would have seen that the young people would have been obviously much higher than the older because they have more muscle mass. So yes, it's all normalized to muscle mass.
Okay, thank you.
Yep. Excuse me. Any other questions?
Hey, Mark.
Hey.
Yeah, so do you have a hint as to why the men and women have a different response to the same training? Like, is it the hormonal status? What is really driving this difference?
We think it's hormone levels, because that's one of the obvious things that's difference between men and women, and they're driving differences in terms of things like, as I mentioned briefly earlier, we've got this regulatory light chain phosphorylation difference that we've seen. There have been studies done in mice that shows you change the levels of estrogen, you change the levels of RLC phosphorylation. And so that's one of the reasons we went after that particular post-translational modification. So yes, and these studies are done in mice. So they take mice, they overectomize them, and then they look and they see this large drop in estrogen, and then large drop in RLC phosphorylation. And then they give them estrogen again once they've overectomized them, and then the RLC phosphorylation comes back up. So yes, we think it's related to hormonal status and different changes that's occurring throughout the muscle, but nailing down exactly which ones those are proving difficult, but we're working on those.
Yeah, you seemed, thank you, Dr. Miller, for this presentation. Clara, I'm a PhD student in the Department of Environmental Health Sciences. So you answered some of my, questions. However, as you suggested that the muscle or the myosin molecule is a sexual dimorphic, it reacts differently in women and men. And earlier in your talk, you talked not just about exercise, changing those, but also drugs. So So I wonder if any pharmaceuticals were tested or environmental chemicals make difference in the performance of muscles and molecules.
Sure, that's a big area of study currently in muscle, and especially in cardiac tissue. So what drugs can we give people to make their cardiac muscle perform better and stop dilated cardiomyopathy and these different things that we see. So yeah, there's a wide range of drugs and different things you can treat people with. It's just figuring out, there's whole companies now that are set up on that. They have a huge path. They can do an enormous amount of experiments in a short period of time and just look to see what's causing changes in these interactions, and they pull out those drugs and then they study them more. So for these studies, we haven't done anything like that. We haven't looked at any drugs. We're just trying to understand what's happening in healthy, relatively sedentary older adults. Did that answer your question?
Yeah, in part. Like, I wonder if you even account for, do you ask people if they take any medication or about their exposure to different environmental chemicals?
So we certainly ask them their health history. We have a detailed health history. We know all the medications that they're on. There's no questions in there about what's been your environmental exposure, for instance, but we certainly know what medications they're on and have looked at different things like that. So there's been some indications that some drugs, for instance, that you take for blood pressure may cause issues with the myosin. And so once we at one point we took multiple studies and lumped them together and had people that were on this medication and people that weren't, and we're able to look and see that we didn't see differences even if they were taking this medication. But there's certainly other things that we've looked at too, same sort of idea. Once we've done multiple studies, we can kind of lump people together. And one of the things that we have seen that does change skeletal muscle function is the level of obesity that you have, or the amount of fat that you have in your body. We are certainly seeing differences there. And so far with our work, we're seeing, again, larger differences in women than we are with men. It seems to be the amount of lipids that you have is more profound, has a more profound effect in women than it does in men. But we also have, that was just kind of taking a bunch of studies and lumping them together. One of the grants that we're currently working on that Professor Kent is a PI for is looking specifically at that as how fat infiltration affects skeletal muscle function from the whole body level down to the molecular.
Hey, Dr. Miller.
Hey, Jay.
How you doing?
Good, and you?
Oh, you know, surviving.
That's good.
I have a question related to your re-enrollment. Do you potentially expect to see bimodal response with participants because of the pandemic? If so, how do you expect to control for it? Because, you know, someone like myself, I'm training a lot for 13, 14 years. So with people who are sedentary, and I changed my workouts completely and respond differently over, you know, just 3 months. So with people who were sedentary or maybe picked up, but then did nothing, or vice versa, you know, physical activity, training habits, do you expect to see any major issues? Because a year, 15 months, it's a long time to do something or not do something most often.
Right. No, it's a good question, but our screening procedure is to talk to people about, you know, the exercise that they've been doing. And of course, we give them the accelerometer and they take that home with them. So if the pandemic made them become, let's say they were relatively active and the pandemic made them become sedentary, they've probably been that way for a year or more and have gone through the changes. And so they'd be good to put in our study. But of course, the reverse is going to be true. I can certainly say I've seen more people walking around my neighborhood during a pandemic than ever walked around before. And so some people may have picked up their exercise level, but if they've done that, then we won't have them in the study. So, you know, as we bring people in, we ask them these detailed questions and try to get a really good feel for their activity levels so that we are starting everyone off at the same level. And if they've been doing it for a year or more, then I think their muscles will have changed and they'll be ready to enter the study. As a funny side note, Jay, I can say that I remember on March 13th when the university closed down, we were discussing, so if we're closed for a month, can we get these people back in the study? Or if we're closed for 2 months, what's the cutoff? Well, now that we know it's over a year, we know that they've lost all their training benefits. There is a question in the chat I'll read. I didn't see this till just now, so sorry, Megan. But the question was, have you compared your sedentary younger adults with more active younger adults or effects of pandemic-related reductions in activity on muscle function? So going along with your question, Jay. So no, we have not compared sedentary younger with more active. Really, my focus has been, the real focus I'm on is the older adults, and the younger adults were almost the control group in that study I'm talking about. You know, how are the older adults changing? So we haven't looked at more active younger adults. And again, we haven't been into this, but there are a number of studies that certainly look at disuse. So you take people, do some studies beforehand, and then you put them in bed rest or something else like that and look at how things change. So there's plenty of studies out there that have looked at how changes in activity are going to change your muscle function. So we haven't done that, but there are certainly interesting questions. Are there other questions anybody had?
Last question. Is there room, do you think, to have a subset of individuals that have finished that maybe didn't continue with training that you could biopsy or test in some way if you wanted a pandemic related aim or add-on or something like that? Just something to think about.
No, it's a great idea. And Jay, we certainly thought about that, and especially pre-pandemic. We were sitting there thinking, okay, we have these people that have gone through the exercise training study. And of course, we talk to them when they leave the study, and I generally ask them, are you going to continue with this exercise? And some will say, no, this was good for me, or I'm going to go on. Yes, I'm going to sign up. I've already signed up for a gym membership. I'm starting next week. What would have been really interesting is potentially takes these folks and look at them 6 months to a year later after our study and ask them, had they continued the exercise, had they not, and look to see how long these changes stuck around for. I would think that the pandemic at this point has kind of thrown a wrench into that, because everything kind of has changed quite a bit due to that. But it certainly is a great idea, and something you'd want to look at is, are people, and even just a general question, are people so impressed with their changes and how much better they're able to perform that they're going to stick with it? And in general, most of the people that are leaving the study are saying they're going to stick with it. Whether they're just telling me that because they think that's what I want to hear or not, I don't think so. I think they're generally impressed with the amount of improvement they've seen and what they've been able to do. And what's nice about these types of studies is most of the people who sign up are pretty excited to do this work because they know they should be exercising more. So they come in with a lot of energy, and they see this as, typically people see this as a way to get themselves kick-started, to get into that exercise program, to get in that routine, and then keep going. No, but that's an excellent point. A follow-up study would be a great thing to do. Are there any other questions?
I don't see any other questions, and I want to be considerate of Mark, your health. I hope you, we do have to ask ourselves, how are we getting viruses in this day and age, right? Given all the precautions we're taking.
My kids did go back to school last week in person, and they seemed completely fine, but I don't know if that's it or allergies or what. But
well, I do hope that you get better, you take care of your throat there. And thank you so much for a very interesting talk. We really appreciate it. And thank you everybody for attending. And for those students who are on, good luck in the last week of classes, and as well as in your exams. We wish you the best. Thank you, everybody.
Thank you.